Hello and welcome to another lecture. Today's lecture is about transcription. So before we start with today's topic, like always, let's take a look back at what we talked about last time. So last time we looked back at DNA and the structure and a little bit of the function of the DNA. And we said that experiments showed that DNA by itself can provide a living cell with information for new acquired functions, and that process is called transformation. And you may recall that we talked about the experiments by Griffith and by Avery and colleagues. I then introduced you to the central dogma, which basically just states that heritable information flows from DNA to RNA to protein. We talked about what it means to that DNA is structured in an anti-parallel way, and that it has complementary strands. And then we looked at the fact that when you have double-stranded, anti-parallel complementary strands, that one can serve as a template for the other in transcription or in replication of DNA. And so today we will talk more about transcription. So that is the formation of RNA in the process of copying information from the DNA into the mRNA. So let's first recap quickly. What are the three major differences between DNA and RNA? We talked about that in the second lecture, I believe. So what are the differences between DNA and RNA? Well, first of all, DNA uses a deoxyribose sugar. So here's a deoxyribose sugar versus a ribose sugar. And remember that this is the deoxy right here because that's just a hydrogen, and there's a hydroxyl group here. So that's the one difference between DNA and RNA use. The second one, of course, is that DNA uses a base that isn't used in RNA and vice versa, and that is thymine versus uracil. So here we have thymine, here we have uracil, and you see how they are quite similar here, except that there's that one methyl group over here in thymine that you don't find in uracil. And then the third one is that DNA is usually double-stranded in the double-stranded helix, while RNA is usually single-stranded. There are some exceptions for double-stranded RNA, but for the most part, and mostly what we will discuss in this class, RNA, mRNA, or messenger RNA, is single-stranded. All right, so let's see how mRNA is made. So that process is called transcription. The DNA is transcribed into mRNA. And so the double-stranded complementary nature of DNA allows one strand to serve as the template for the other. And so therefore this one here is now referred to as the template strand. That is what provides the template for the RNA as it's being produced. And the other thing that you see here is that the mRNA, the RNA transcript here, starts at the 5' end, and then nucleotides are added on successively from the 5' end to the 3' end. And so that is referred to as the RNA running from 5' to 3'. Now note that if you look at the DNA, the 5' end is over here, right? And the 3' end is over here. So when we say 5' to 3' is the direction of synthesis, then what we mean is that the new molecule is synthesized with its end, its start, being the 5' end, and its latest attached nucleotide being at the 3' end. And then, of course, if you look at all these polarities, as they're called, the numbers, the 5' and 3' ends, that's the polarity of the DNA strand, not to be confused with polarity in a molecule due to charges, right? So this is the polarity of the strands. You have 3' to 5' here, 3' and 5' here, and you have 5' here. So they're always opposite to each other, right? That's why they are complementary and why they are anti-parallel. All right, so now we have our DNA being opened up. We have RNA being produced starting at the 5' end, going to the 3' end. And transcription is always initiated at a region in eukaryotic genes. We'll talk about what eukaryotes and prokaryotes are in just a few minutes. So in those types of cells that have a nucleus, eukaryotes, they have a promoter where transcription starts, and then it's synthesized in the 5' to 3' direction. And then it ends, transcription ends at the terminator. Now, there are 2 strands to the DNA, and it doesn't really matter which strand serves as the template. For this particular gene, for this transcript, that bit serves as the template. But it's also possible that the other strand can serve as the template. So you see here, in this case, the lower strand serves as the template for this particular gene. And so again, RNA is transcribed from 5' to 3', but since it's the lower strand and that strand has the other polarity, the other direction in its antiparallel structure, that gene would have to be transcribed from 5' to 3' in this direction. So 5' to 3' in this direction on the lower strand serving as template, upper strand serving as template, 5' to 3' direction in the other direction. Okay, so now that we've talked about in which direction transcription goes, let's take a look at what is required for a strand of DNA to actually suddenly become actively transcribed. Most of the time in a cell, the DNA just sits there. Most genes are not transcribed at any given time unless or until they activate it. And so that activation occurs by binding of a number of proteins, and many of these are called transcription factors. So let's take a look here at this strand of DNA, and there's a promoter and there's the transcriptional start site, right? So it starts at the promoter. In fact, it starts a little bit after the promoter, but let's just say the promoter is the place where transcription is initiated. And then there are these proteins that are called transcription factors, just, proteins that serve as a signal, or they help as a signal to start the process of transcription. And they're just called transcription factors because they act to allow transcription to occur. And they're called general transcription factors because many of these transcription factors are the same for all genes, but then there are also specific transcription factors which are only for specific genes. But for right now, let's just look at the transcription factors, a number of proteins that bind to the promoter. So the promoter is a piece of DNA, and general transcription factors are proteins that bind to that piece of DNA. Now, that gene might be transcribed in this direction, and upstream, as it's called, upstream of it are regions on the DNA that are called enhancer sequences. They enhance the transcription or the transcriptional activity of a particular gene. It's possible that the gene can be transcribed without the enhancers, but not very strongly. And so the enhancers enhance the transcription, and they require the binding of proteins to these enhancer sites that are called transcriptional activator proteins. And in order for these proteins to act together with these general transcription factors to make that gene really strongly active, they have to bind together. And so they loop around this piece of DNA. This protein and this protein can bind to this entire complex of proteins. Here are general transcription factors. And then there's a set of so-called mediator complex proteins. And they all together, then this entire set of proteins is required for transcription to occur. And then the last important bit here is, after looking at the general transcription factors and the mediator complex, all proteins here in this purple blob, and the transcriptional activator proteins, is this green set of protein complexes, and that's called polymerase II, or RNA polymerase II. It makes RNA, polymerizes RNA, mRNA, in this case for polymerase II, from individual nucleotides. So that entire region here, of this entire set of proteins in this region, is required to be set up just right for transcription to be able to start. So there's one thing that often gets confused, and that is that, I just gave you a bunch of new names to learn about. Some of them refer to proteins and others refer to the DNA regions. So I sort of wanted to separate that here so that you can be absolutely sure what is what. There are terms that refer to the regions on the DNA, and that's the enhancer sequence, the promoter, and the transcriptional start site. These are DNA sequences, A, T, C, and G. This is bits of DNA, certain bits of DNA. To these bits of DNA is where the proteins bind, right? So these are made from amino acids. These are long proteins that have a certain function, many of them enzymes. And so the following terms refer to the proteins that bind to this DNA. That is transcriptional activators, transcription factors, the mediator complex, which is made up of many proteins, and then the RNA polymerase too. Right? So make sure that you distinguish between terminology that refers to the DNA versus terminology that refers to the proteins. Not so fast. Okay, so let's zoom in a little bit onto the RNA polymerase. So this is now our RNA polymerase, and all the transcription factors are not shown in this particular slide. We have the DNA, it's double-stranded, and we have the RNA. The RNA is being made, and so you see that this this polymerase, that's the green part, made up of multiple subunits, that binds around the DNA, and inside this protein complex, there are regions of the protein that allow the protein, the RNA polymerase, to sort of, like a thumb, if you will, sort of hinge in between the two DNA strands and pull it apart just a little bit, so that RNA bits can be added and can produce this, what's called a nascent strand. Nascent, from the Latin word being born, is what it means. So this is the RNA that's being born, that's being made, currently produced. And you see that it started at the 5' end, and here at the 3' end is where new nucleotides are being added. And this entire region here is called the transcription bubble, because it sort of bubbles open the DNA, and the RNA is being made there. So inside the transcription bubble is where transcription is occurring with the help of RNA polymerase by adding nucleotides to the 3' end of the growing mRNA strand. Okay, so just so that you have it in writing here as well, transcription factors, mediator complexes, and all that allow the transcription to start. That's called transcriptional initiation. The DNA then separates, and then RNA polymerase number 2. There are other RNA polymerases. That's why it's called here RNA polymerase 2. Reads the transcript and then adds the complementary nucleotides on. Okay, so this bit is called transcriptional initiation. So once the RNA polymerase is in full swing, it just keeps chugging along the DNA and it keeps adding stuff. And as it's doing that, it elongates the RNA. And so that's called the elongation process, transcriptional elongation process. And so during that process, what you have is that incoming ribonucleotides are accepted if they are correctly base pairing with a template. So if this is our DNA template strand, there has to be a check, and that's the RNA polymerase that does the checking. Checks if this is the correct correct formation of the A with a U or G with a C. And of course, here you see the hydrogen bonding. The 3' hydroxyl group of the growing strand attacks the high energy phosphate bond of the incoming ribonucleotide, providing the energy to drive the reaction. And what that means is that this set of 3 phosphates right here, that bond, these bonds between phosphates, is very energy rich. And when you split a bond, that energy is liberated. And so that energy can then be used for the formation of the new bond. And so the 2 phosphates of the incoming ribonucleotide are then released. These 2 phosphates are released as pyrophosphate. And then that phosphate here becomes the next link. So that process is called transcriptional elongation. So basically the chugging along of the RNA polymerase along the DNA, adding more nucleotides to the strand. Eventually, there is termination, and termination, there are certain termination factors, but your book doesn't go into much detail of that, and so I won't either. What your book does talk about, though, is that there are slight differences, yet many similarities between the way that eukaryotes and prokaryotes do transcription. And so that's interesting for a number of reasons. One is that prokaryotes are mostly bacteria. There's lots of bacterial types of cells, organisms, and others that are referred to as prokaryotes. And then there are the eukaryotes, and the eukaryotes are, what we usually think about when we think about animals or plants. So exactly what are prokaryotic cells and what are eukaryotic cells we will discuss in the future. But since your book talks about the similarity between the transcriptional process of prokaryotes and eukaryotes, let's just spend a minute or two on looking what the differences are between these two types of cells. So we have a prokaryotic cell, as I said, so for all intents and purposes, we can think about bacteria here when we talk about prokaryotic cells, and when we talk about eukaryotic cells, we think about animals and plants. They're called eukaryotic cells, eu meaning real, and karyon means nucleus. So they have a real nucleus, and that is really the main difference between a eukaryotic cell and a prokaryotic cell. So prokaryotic cells have a prokaryon, a pronucleus. It's not a real nucleus, and real is here defined as having a membrane that is surrounding the DNA in the nucleus. And there's something here, the DNA sort of sticks together in what's called a nucleoid, but it's not a real nucleus. And so it's a prokaryotic cell, not a eukaryotic cell. So that is one of the main differences. Prokaryotes also have cell walls. Eukaryotes may or may not have cell walls. So the animal cells do not have a cell wall. The animal eukaryotic cells do not have a cell wall, while plant cells do. So that's not the one distinguishing factor between prokaryotes and eukaryotes. But in eukaryotes, since the nucleus is membrane bounded, there are other membrane bounded organelles that are also in the eukaryote, and we will talk about those in a little bit later on in a different lecture. But you may have heard about mitochondria, you may have heard about chloroplasts. So those are things that are found in eukaryotic cells, but not in prokaryotic cells. All right, so back to our transcription and what your book describes here as the difference between transcription in prokaryotes and eukaryotes. So basically, what you might want to know about the differences in these processes is that the RNA polymerase II in prokaryotes looks very similar. So this is a cartoon of the RNA polymerase in a prokaryote. The other one that I showed you was a eukaryote. Similar here, again, you have the bubble, you have the addition of the incoming RNA nucleotides, you have the growing RNA strand. All of that's very similar. But the differences that you can find between prokaryotes and eukaryotes in transcription is that prokaryotes require fewer proteins for initiation. So all those transcription factors that I showed you, there are some factors that are required for transcription, but in general, fewer. There's something that's called the sigma factor that is used in prokaryotes, and that's kind of a neat transcription factor, if you will, because prokaryotes can just use that sigma factor, and there are different types of sigma factors, but they can just use one sigma factor, and that will start the transcription of a whole slew of different genes. So prokaryotes use sigma factor transcription factors, and they are unique to prokaryotes, not eukaryotes. And then the prokaryotic RNA polymerase transcribes multiple genes at the same time. So there's basically, a certain process that needs to happen, maybe metabolism of a certain sugar that suddenly is around, and the bacterium sees that this sugar is around. So now it needs to break it down, it needs to cut it into pieces, it needs to make all kinds of smaller bits out of this sugar in order to metabolize it. And so multiple genes are necessary. And so rather than having transcription factors that are different for each different gene that is now dealing with this new food source, they just transcribe them all at the same time. And so that is another difference in the transcriptional process between prokaryotes and eukaryotes, that prokaryotes, they transcribe many genes at the same time. Eukaryotic polymerase usually just one. So that's what we have here for the difference between prokaryotic and eukaryotic transcription. The other thing that I want to point out that's also different in prokaryotes is that as soon as the mRNA is made, translation occurs. So we talked earlier about the dogma, the central dogma, DNA, RNA, protein. And so the process of making RNA from DNA is called transcription. We're talking about that right now. The process of translating that mRNA into protein, that's called translation, and that occurs along these ribosomes. And we'll talk about those in more detail later. But that happens at the same time as the transcription still occurs. So this is not even done yet when the ribosome already starts making the protein. So when it's already translating. That's another difference that your book points out. Okay, so for the rest of the lecture, let's talk about eukaryotic transcription in just a little bit more detail. So there are some things that are just more involved, sophisticated, more complicated, if you will, in eukaryotic gene transcription. And so a few things I want to point out, and those are some things that I'd like you to know about. So this thing here is a eukaryotic cell, and it just shows that there's that, that cell, that nucleus that has the cell inside. And so that's what they're pointing out here. So they take a little bit of DNA out of a eukaryotic cell. On the previous one here on this slide, here was our prokaryotic cell, right? So that's how the book makes the difference. Okay, eukaryotic cell, DNA, and that DNA is transcribed into RNA. So far, so good. We've talked about that before. What I want to point out in this particular figure is that the DNA has parts within a gene, and we haven't defined the gene yet, I know, but the gene makes eventually a protein, and the protein has a certain function. But a gene being made of DNA, in fact, in eukaryotes, has intervening regions called introns that are not needed for the protein. I'm sort of shaking my head a little bit because that's not completely true. They're not completely useless. There are certain ways how introns are used, but for all intents and purposes, really the most important thing to know about introns is that they get usually removed. So they get spliced out, they get thrown away. The exons get kept. And so this is what this figure shows. Exons stay, introns get removed. And so in order to take out all this stuff that was transcribed in the transcription process here in what's called the primary transcript, see, they all still contain these introns that eventually have to leave. In order to get that out of the primary or immature RNA, splicing occurs. So splicing is just like, you basically take a piece of string and you cut little pieces out, and then you fuse the rests together that you want to keep. That's the splicing process. That's what happens, and then you get a mature spliced mRNA after that process. And then there's a few other things that happen. At the end of this spliced mRNA, there are two bits that are added, and they help with stability of the mRNA. Otherwise, mRNA is very unstable, can degrade very, very rapidly. DNA, by the way, is not. DNA is very stable, but mRNA is very unstable. So in order to make it more stable, a so-called 5' cap is added. It's a has a molecular structure. I'll show you in a second. Not so important that you know the structure, just that you sort of know what it is in general. And it's at the 5' end. And then at the 3' end, it's called a poly-A tail. That's another structure that allows for the mRNA that's in between to be safe from enzymes that just sort of degrade RNA all the time. The poly-A tail is just nucleotides, a bunch of nucleotides that are all A's, and the cell knows what to do with that. The cell knows that that is the end. It's not to be translated into any protein. We'll talk about that also later. So, okay, we have the cap, we have the tail, and then we have the splicing of the exons where the introns are removed. So I always try to remember that by saying introns are the intervening, the intervening pieces that are getting spliced out. Okay, so that 5' cap looks like this. Here's our RNA, right? Here's our base, our sugar, our phosphate. That's the 5' end. Here's 3', 5', 3', 5', and so forth. The 3' end is on the bottom. That's not what we're interested in right now. Here, this is our RNA. What we're interested in is the 5' cap. This here is the 5' cap. So your book says the 5' cap consists of a modified base, that's this, linked by its 5' carbon to the 5' end of the primary transcript by a bridge composed of 3 phosphates. So this sugar part here, this is basically our base, right? That we have seen before. It's guanosine. It's basically G, but it has an extra methyl group here. So that's why it's called a modified base. But it's essentially just another G on there that isn't recognized as a G because of that methyl group. And that's linked with this with these 3 phosphate groups here to the end. That gives you the 5' cap. That makes it stable. Okay, so let's look at the splicing process in a little more detail. How does splicing occur? Well, splicing occurs because there are proteins, these little blue balls here on your figure. They are proteins that act with each other. There's other molecules as well, and they together produce what's called the spliceosome. So the spliceosome is a set of molecules, many of which are proteins, that bind at certain regions. They bind only at regions that are indicative of the beginning of the intron and indicative of the middle of an intron. There is a certain set of nucleotide sequences in the middle of an intron that can attract this protein. And when that protein is around, and these proteins are around, and they're binding at the end, then they can together form this loop or lariat, and then pull this end towards this end, take out the pink part. That is now our intron that gets degraded, and then the spliced exon is what the spliced exons next to each other are then what's left. So if you have a gene with multiple exons and multiple introns, there are multiple ways how you can splice this together. Not too long ago, I would say 10, 20 years or so, people thought that all the introns have to leave and all the exons stay. And now you already heard me a little waver there earlier on that, well, introns mostly leave, and hatching my bets here a little bit. Reason why I'm doing that is because there is a process that's called alternative splicing, and we've only learned about that in the last 10, 20 years or so. We've only learned how important of a process that is in that time period. We kind of knew that that could happen before. Okay, what happens exactly? Alternative splicing means that sometimes all the exons are used and all the introns are not. Sometimes some exons are used, but others aren't. So in this example, exon 1, 2, and 4 are used, spliced together. And in others, 1, 2, and 3 are used, but not 4. So you can have a mature mRNA that has different parts of the gene. So this one has exon 1, 2, and 4. This one has exon 1, 2, and 3. And you might imagine that there might be one that has only exons 1 and 4. And of course, when that bit gets translated into protein, and that bit gets translated into protein, that will give you a different protein, because either it has this piece, or doesn't, right? So it's a longer or it's a shorter piece of mRNA that's translated into protein, leading to shorter or longer bits of protein, and also different types of amino acids that get translated into the protein. We haven't discussed that detail yet, so no worries if that still sounds a bit nebulous. But what you should be able to understand already is that if you have different parts of the gene that are spliced together to produce a mature mRNA that's producing the protein, that's got to be different than if you have a different set of pieces of DNA. All right, so that's alternative splicing. And with that, we're already at the end of the lecture. And the summary for today is that eukaryotes have membrane-bounded nuclei. They have a nucleus, and prokaryotes do not. So that is one important take-home for today. Eukaryotic transcription starts at the promoter and ends at the transcriptional terminator. That's important to remember. RNA is synthesized in the 5' to 3' direction. And we talked about where we find those numbers and why those numbers are called what they are called. Transcription requires the binding of transcription factors and the RNA polymerase. Otherwise, transcription won't start. Eukaryotic mRNA matures after transcription, and that maturation process is comprised of the 5' cap addition, the 3' poly A tail, and then the introns that are spliced out. And then we talked at the very end about alternative splicing and how from one gene you can get multiple mRNAs, mature mRNAs that can produce different proteins. And with that, we're at the end of today's lecture. I see you in class. Bye-bye.